Okay. The material and color stream research aims to catalog unknown dye molecules to determine their usefulness for photopolymerization reactions. Here we present preliminary cyclic voltammetry characterization of photoredox catalysts called PRCs. These are the key molecular components in photopolymerization. Photopolymerization occurs when PRC is excited by light, which initiates the polymerization reaction to create a polymer from a liquid resin. We can estimate the PRC's energy using cyclic voltammetry. Understanding the energetic redox landscape of these molecules allows for a more rational choice of resin components for successful polymerization. The experimental data of PRC or dyes collected will be used for a later application.
Electrochemistry is a subfield of chemistry focusing on the relationship between electrical potential differences and the flow of electrons or the current. Cyclic voltammetry, or CV, is an electrochemical experiment where the system's potential is controlled and the current flow is monitored. A solution is exposed to a varying electrode potential, and the electrical current that flows in response is observed and analyzed. The CV experiment aims to determine whether a molecule is redox reactive. The redox potential is the energy required to drive the reaction, and it is important for matching the energies of resins for optimal photopolymerization reactivity. Polymerization is the chemical process of combining individual monomers to form a 3D polymer. Resins are composed of PRCs, monomers, photoinitiators, and co-initiators. Co-initiators help drive the polymerization reaction by donating or accepting electrons. Our initial CV experiment demonstrates the expected co-initiators' redox potentials.
First step was identifying co-initiators of interest. The two co-initiators used in this experiment are HNU254 and EDMAB. We verify the shape of these peaks using CV and ascribe the redox potentials of these co-initiators. The electrochemical cell is made out of glass with a cap to fit 3 electrodes. The solvent contains an electrolyte which allows the current to pass through the solution. Once the cell is prepared, we add ferrocene as internal standard to center the electrochemical window. The peaks from the CVs were then analyzed for further investigation.
HNU254 is a known oxidizing agent, meaning reduction peak should be seen in figure 8. HNU254 has a well-defined reduction peak at -3 volts. There is a ferrocene peak at top right of figure 8. Conversely, EDMAB is a known reducing agent, an electron donor. EDMAB has an apparent oxidation peak at around 0.77 volts and no reduction peak, indicating an irreversible reaction. There's also a ferrocene peak at 0 volts, as shown in figure 9. CVs help to validate co-initiator redox potentials. We can also better understand the molecule's energetic landscape, helping us choose better resin components for future photopolymerization reactions. Future investigations in electrochemistry include expanding the CV experiment to unknown dye-based PRCs and combining qualitative polymerization reactions with the CV reading. Thank you for listening to our presentation.